Welcome to Regents Review for your Living Environment Regents. This is Ms. Winans, and today I will cover Session 3, Cells, Transport, and Biochem 1. Today we're going to start with cell biology. In order to study a cell, you need a microscope. The discovery of the microscope, the invention of the microscope was imperative to our understanding that life can exist even in the microscopic world. So for a very long time, unicellular organisms existed, but nobody knew about them. It wasn't until the invention of the microscope that allowed us to start to see a whole new world of living organisms that we didn't know existed. In class, you use the compound microscope, which is the microscope that's on this slide. You will not be asked to label the parts of a microscope on the Living Environment Regents. However, there are 2 New York State labs that use the compound microscope, so therefore it is fair game for Part D of the New York Living Environment Regents. So we're just going to take a look at some important parts of the microscope. So we have the ocular lens, which is in your eyepiece. Yours in class had a magnification of 10x. You also have the objective lenses, which are right here on the nose piece. This has the additional lens. So when you use your compound microscope, there's going to be a light source down at the bottom. That light source is going to pass through the slide into the objective lens and then through the ocular lens. Since it passes through the lens, it passes through 2 lenses, that's going to cause the image to appear upside down and backwards. You'll get more into detail as to why that happens when you take your physics classes. The objective lenses, you had a scanning objective lens, which was 4x. You had a low power objective lens, which I think was 10x. And then you had your high power objective lens, which was 40x. Okay, so these are the pieces that you really want to focus on. So you have your ocular lens, you have your 3 objective lenses, you have your light source, and you have your slide. Again, the light source is going to pass through the slide, then to the objective lens, and then through the ocular lens. Again, that's going to cause the image to be upside down and inverted, which is what you saw with the letter E during your lab. Whenever you're determining magnification, you're going to take the ocular lens, which in this case is 10x. Right now it looks like we're using the objective lens, which is 40x. You multiply the 2, so the image will be 400 times greater when using the objective lens, when using this microscope. Again, the microscope we used in class is called the compound microscope, and scientists are going to use the compound microscope to look at cells and microscopic organisms that are unicellular. As time went on, science really expanded their exploration of cells and cell biology. And in order to get more information, technology had to advance as well. So the invention of the electron microscope enhanced our understanding of cell biology because it allowed us to now not only look at cells, we now could look at the tiny structures found inside the cells called organelles, which actually means tiny organs. So with the electron microscope, we are now able to see the details of the organelles. So compound microscopes are going to be used to analyze details of cells. Electron microscopes are going to greatly increase the magnification and allow us to study the details of organelles. If we take a look at this picture here, we see the Elodea leaf. This is a picture taken by one of your classmates during the Animal vs. Plant lab. The Elodea leaf is part of a plant, so you'll notice that each one of these boxes is one plant cell. So we can see actually many plant cells. We can see the cell wall around the outside, and we can see the chloroplasts. Okay, so this is an image that you would see with a compound microscope. To compare the images of a compound microscope to the images of an electron microscope, this one structure is one of these tiny little chloroplasts. So again, the purpose of the compound microscope is to allow us to look at the cells. The electron microscope is going to allow us to look at further detail, increase the magnification, and now look at the details of the actual organelles. So, one concept that you need to understand when using the microscope is the concept of field of view. Right now we're taking a look at Google Earth. We can see the entire Earth. So our field of view is very large. We can see the whole planet. We actually can't see the whole planet. We can only see the United States, Canada, Alaska, parts of South America. So this is our viewpoint at this point. So what I'm going to do is I'm going to increase the magnification. So I'm going to type in Limbrook High School, Union Avenue, that's us. And what's going to happen is the magnification, once I click search, is going to increase. As the magnification increases, we're going to notice the field of view, the amount of stuff we can see, decreases. So we can see a lot more detail right now because we can see the high school, we can see the field next to the high school, we can see the kindergarten center, the teacher's parking lot. So we can see a lot more detail. So magnification allows us to see more detail, but it definitely decreases the field of view, the amount of stuff that you can see. So as magnification increases, field of view decreases. I can intensify that by continuing to zoom in. So now I'm decreasing the field of view, but I'm increasing the magnification. Students have the most difficulty when going from scanning to low or low to high. The biggest mistake they make is they skip these two important steps and they try to take a shortcut and skip all the way from scanning to high power. Remember when we took a look at Google Earth, we said that the field of view decreases as the magnification increases. So the amount of stuff that you can see on the slide is going to reduce. So if I was focusing in on this little dot over here, I can see it under scanning power, but if I don't center it before I switch to low power, it looks like I lost my image. So it's really important that when focusing a microscope, you remember that you have to center the specimen each time you increase the power. And you must go in order. You can't skip steps. You have to use the scanning power, use the coarse adjustment knob, fine adjustment knob, low power, coarse adjustment knob, fine adjustment knob. Under high power, please only use the fine adjustment knob to prevent the objective lens from slamming into the slide.
Hi, I'm Miss Pryor, and my salt experiment is diffusion through the red onion. Take the slide. Take a sample of red onion skin, like so, and place it on the slide. Then take a couple drops of water and add it to the slide. Take a cover slip and hold. Let's focus the microscope. Place the slide on the microscope under the scanning objective lens. Use the coarse adjustment knob and fine adjustment knob to focus the image. Switch to low power, use the coarse, then fine. Once focused, switch to high power and only use. Let's take a look at our onion cell. This is an onion cell in regular tap water. Notice the placement of the cell wall and cell membrane. Many students have difficulties determining how to add salt water to an onion cell. Which picture illustrates the best way to add salt solution to an onion cell? Pictures 1 and 4 do not have anything to do with this lab. We can eliminate the choices automatically. Choice 3 tells us to shake salt directly onto the leaf, or in this case, the onion cell. That is not the proper way to add salt to an onion cell. Picture 2 is the best way to add salt to an onion cell. Now we're going to add salt to the slide. You drop the salt water on one side of the slide. Then gently dab on the opposite side to pull the salt across the slide. Now let's take a look at our cells under salt water solution. Notice the location of the cell wall and the cell membrane. The cell shrinks in the presence of salt water.
The last thing we're going to talk about in terms of microscopes is microscopic measurement. So images under the microscope are way too small to use a millimeter for measurement. So what we use is what is called a micrometer, which units are represented by UM. For every 1 millimeter, there are 1,000 micrometers. So if we were to write this in scientific notation, 1,000 is 1 times 10 to the third. So how many micrometers is one cell? So on the living environment Regents, they may show you cells and you have to estimate the length. Sometimes you're going to have to measure the length in millimeters and convert to micrometers, or measure it in micrometers and go back to millimeters. So in this case, we have 2 cells, and they're telling us that 2 cells are 1 millimeter. So 1 cell would be half of that, so that would be 0.5 millimeters. So it says that 1 millimeters is equal to 1 times 10 to the third micrometers, which means that we would move the decimal point 3 points. So we have 1, 2, 3, resulting in 0.5 millimeters or 500 micrometers. There are exceptions to this cell theory. Remember, the cell theory states that the cell is the basic unit of structure and function in living organisms. All living things are made up of cells, and cells come from pre-existing cells. That being said, there are some things that exist in life that we can't really figure out if it's living or not if we use the cell theory as the definition of something that is living. Another component of a living thing besides a cell is that the cell has DNA. DNA is the hereditary material that makes up the genetics or the instructions to make the protein. And viruses do have DNA, but they're not made up of cells. If we look at the animation on the bottom, we'll notice that these little viruses are docking on the surface of the cell membrane. When they dock on the cell membrane, they're injecting something into the cell, and it's causing the viruses to burst out. This is how they reproduce. Viruses depend on a living host. For example, this is a bacteriophage. They infect bacteria. So the viruses dock on the surface of the cell membrane. They inject their DNA. The DNA is inserted into the host DNA, and now the cell only knows how to make baby viruses, causing the cell to die, but the viruses to reproduce. So the virus is a complication to the cell theory because it has DNA, but it can't reproduce on its own. It requires a host. So therefore, the virus is an exception to the cell theory. So there are different types of cells. Cells in an individual have identical DNA. We all know that cells in other individuals have different DNA. We can see it. When we look at each other, we notice differences. But inside an individual, the cells in that individual have the same DNA. However, we have tons of cells in our body that carry out very specific functions. For example, we have nerve cells, white blood cells, skin cells, muscle cells. There has to be a point in which we assign them a job. Think of your lab manual. When you get your lab manual, you're not going to read the entire lab manual just to do one lab. You're going to read that one specific lab, carry out all the tasks for that specific lab, and gain the knowledge that you were supposed to gain from that activity. So at some point, we need to change a cell that has access to all the DNA, which is called a stem cell, into a cell that only has access to certain parts of the DNA, which is called a differentiated cell. The way we do that is we turn certain genes on and off during development. This process is called differentiation. So if we look at the animation below, we notice that we're going to be a nerve cell. Nerve cells are called neurons. They function in our central and peripheral nervous system. What we're going to do is we're going to turn all the genes in the DNA to be a nerve cell on, and we're going to turn all the genes to be all the other cells off. This has now differentiated the cell. So while the baby is developing in the womb, it's going to receive cells that have specific jobs so that we can do a better job at maintaining homeostasis. The cell membrane is one of the most important organelles, and that's why you'll see it in prokaryotes, eukaryotes, animal, and plant cells. The cell membrane controls what goes in and out of the cell. It also handles cellular communication. The cell membrane is composed of two major components. You'll see two layers of fats, which is called the bilipid layer of phospholipids, and then you'll see scattered throughout proteins. These proteins can be peripheral proteins, meaning they're located on the outside of the cell membrane, or they could be integral proteins, which means they act as tunnels to allow things to transfer in and out of the cell. One of the important roles of the cell membrane is communication, and they do this using what are called receptor proteins. Receptor proteins are these things that look kind of like bird feet in these animations. And protein function is determined by shape. And it's very important that we're able to send certain signals to certain cells, but not communicate with other cells. For example, if we want to send a signal to a nerve cell, we probably don't want to send that same signal to a neighboring muscle cell. So we use the shape of proteins called hormones to communicate from cell to cell. And if we don't want to talk to one cell, then we just make sure that the shape of the message only matches the shape of the receptors of the cell we're trying to communicate. So if we look at this picture over here, we notice that cell A can send signal 2, which is in the shape of a triangle. And you can see that this triangle fits inside this receptor protein. Cell B can send signal 1, which is a circle, which means it can communicate with cell A because it matches the shape of its protein receptor. However, if there was a neighboring cell that had the same function as cell B and had the same protein receptors, cell B would not be able to communicate with this cell because it sends signal 1. Signal 1 is circular, and this additional cell is only sending out protein receptors that are triangular shaped. So this is a unique way in which cells can communicate with each other and ensure that the messages that they send go to the cells that they want to talk to. Another important role of the cell membrane is cellular transport, letting things go in and out of the cell. The cell is what is called semi-permeable. It only lets certain substances in and out of the cell. And one way it controls what goes in and out of the cell is using a concentration gradient. A concentration gradient is how much of a substance is on the inside of the cell versus outside of the cell. In passive transport, molecules will naturally flow from an area of high concentration to an area of low concentration. Whenever you're looking at a transport question, you always want to identify the concentration gradient. So if we were to count the dots, I could see that A is the high concentration and B is the low concentration. So I always tell students to think of a party. If you have a bunch of people and you have them in your basement, and they're over in the basement and it gets crowded and crowded and crowded, what happens, maybe you open a window, but it's really congested in there, and people don't like to stand on top of each other. So if you were to go to your basement door and open it up, people would naturally flow to the outside area, because just like molecules, people don't like to be crowded. So this is a natural movement from a high concentration to a low concentration. This is called passive. It requires no energy and will happen naturally. If you spray an air freshener in one side of the room, eventually those molecules will spread out to the entire area because of the fact that molecules don't like to be crowded. The molecules will stop moving for the most part, even though they will go back and forth, going back to the idea of dynamic equilibrium. It's never going to be at perfect balance, but the molecules will continue to move until both sides of the concentration gradient are relatively equal. If we go back to the party scenario, if it gets a little rowdy outside and there's a noise complaint, you might have to force the people to go back inside. This is going to require a lot of energy because we're now going to be pushing molecules into an area where there's less space. This is active transport. This is when we have to move molecules from a low concentration to a high concentration. We need to force these molecules to go against where they're comfortable. They need to go into a crowded area. The most common area where this happens is in a root of a plant. The soil may be poor in nutrients or water, but what has to happen is they still need to get into that plant. So the plant is going to have to use force, and it's going to have to require energy, which is ATP. Lucky for us, we can see the letters A. Trying to get my marker to work. A, T, P in active transport. So we're going to have to use that energy to force the molecules from the soil into the plant. So active transport is going to use ATP, go from low to high concentration. Passive transport doesn't have to use ATP and is going to go from high to low concentration. Whenever you have a transport question, they're either going to use the word transport or movement of molecules. You always want to determine where the concentration gradient is. So for oxygen, which are represented by the dot, it looks like the low concentration is on the inside and the high concentration is on the outside. And for water, which is represented by an X, it also looks like we have the low concentration on the inside and the high concentration on the outside. We also see arrows indicating that the X's, the water, are moving into the cell and the oxygen is moving into the cell. So if we follow that, we have a movement from high to low high to low concentration, and what that means is that the water molecules are using passive transport. So they are moving from an area of high concentration to an area of low concentration, and the molecules will stop moving once they've reached dynamic equilibrium, meaning there's a relative balance on both sides of the membrane. Okay, to understand biochemistry is to understand how we get the substances we need and how we metabolize them in our cells. So popular thing before a sports event is a pasta party, and there is science behind the pasta party. So when you go to a pasta party, you eat pasta. Pasta is a starch. So it is a polymer. Now, polymers of carbohydrates are ways that we obtain energy. So what people do before a race or a competition is they carbo load. They make sure they have a lot of carbohydrates so they have an energy reserve for the intense competition that they're going to participate in. But polymers are too big to diffuse. They cannot diffuse until they are broken down or digested into their monomer form. So the first thing we have to do is we have to eat the pasta, and that happens through our digestive system. That is going to break down through mechanical and chemical digestion the starch, which is a polymer, into a monosaccharide or simple sugar, which is a monomer. Once it's in its monomer form, it can diffuse in our bloodstream, which is signaled by the red arrow. Remember, diffusion is a type of passive transport. It's moving from an area of high concentration, which is our small intestine, and the low concentration, which is our blood. Our blood is then going to be able to transfer those simple sugar molecules to our cell, where they can interact with our mitochondria. Mitochondria can go through cellular respiration and generate ATP, which we will need during our intense competition the next day. We do the same thing when we eat protein. For example, people who are trying to build muscle mass may increase protein in their diet. That's because protein contains amino acids, and we can take those amino acids, and when we lift the weight, we damage our muscles. We repair those muscles, and when we repair those muscles, they have added structure to them, so we can build muscle mass. So people tend to eat a diet that's high in protein. So what we do is we take the protein, which is a polymer. Again, we digest it. Notice that it's a different shape because it's a different structure. We break it down into amino acids. The amino acids are then transferred through our bloodstream because they can diffuse. Amino acids are monomers. Again, they can diffuse into our bloodstream. They're small enough. And then they can go to our cell where our DNA provides instructions to our ribosome to make human protein.